What's good, y'all? Aki the Scientist here, finally back with another video. This is going to be the start of a new series where we dive into everything biochemistry. Let's jump into it. In this video, we'll be talking about protein structure. Now, I'm sure you've heard of proteins, you eat them every day, but they're not just some food on your plate. I'm going to change the way you see them. They're actually one of the four main macromolecules, including lipids, nucleic acids, carbohydrates, and the topic of this video, proteins. To understand proteins, you need to know what they're made of. So if we zoom in on a molecular level, we see that proteins are these folded chains of monomers or units of amino acids connected by a covalent peptide bond. These three-dimensional molecules are made up of key elements consisting of an amino group, a carboxyl group, and what we call an R group or a side chain. Now, among all the possible amino acids, we typically only see around 20 found in proteins. And the way we distinguish these amino acids are through these variable R groups, or side chains. Here's a list of the amino acids we often work with in biochemistry. It's probably worth noting you'll need to know their abbreviations and structures at some point, so it's definitely worth your time practicing and testing yourself using a whiteboard or a piece of paper. A key tip here is grouping them together based on their R group. For the most part, you'll find some that are acidic, basic, polar, and nonpolar. As I previously mentioned, amino acids are linked together by covalent peptide bonds. These bonds are formed through a condensation reaction, connecting the alpha carbon of the carboxyl end of one amino acid to the nitrogen of the amino group in another amino acid. Now, although these bonds are typically written as a single bond, due to resonance, they take on double bond characteristics. This is critical to remember as this limits the rotational freedom of this part of the peptide chain. And that stereochemical restraint is what determines how the protein backbone can fold. We can think about the three-dimensional nature of proteins by breaking them up into four levels of protein structure. The first, or primary structure, being the sequence of amino acids, followed by the secondary structure. This level is the arrangement in space of the backbone atoms in a polypeptide chain. Two kinds of secondary structures that occur frequently in proteins are the repeating alpha helix and the beta pleated sheet, or simply just called the beta sheet. In contrast to the covalent peptide bond we see in the primary structure, the secondary structure is bound together by hydrogen bonding. The alpha helix and the beta sheet are not the only possible secondary structures, but they are by far the most important and definitely worth going into more. Peptide conformation at the secondary level is described by phi and psi angles, also called Ramachandran angles. You can see the backbone take on a helical shape as the carboxyl group of one amino acid hydrogen bonds with an amino group 4 residues down. Other structures are found in proteins, and they sometimes break up the irregular nature of the motifs we've mentioned just now. Here are some examples of these alternate structures, which differ in the amount of residues per turn and the number of atoms in the ring formed by hydrogen bonding. Also, you might see beta bulges found in anti-parallel beta sheets. I'm not going to go into further details on these, but I wanted to at least mention them and note that their occurrence is a result of steric spatial reasons. Additionally, as we see these secondary structures start to combine, we end up seeing super-secondary structures, such as the beta alpha beta unit, the alpha alpha unit, the beta meander, and the Greek key. One thing I'd like to point out and get your opinion on these secondary structures is their shape. To me, an alpha helix looks like a coil, and the beta sheet sort of reminds me of a separator dielectric in a capacitor. My question to you is, what kind of parallels do you think we can draw from their resemblance to electrical hardware? How might these structures contribute to proteins' role in various biological processes, such as catalysis, signal transduction, and molecular transport? Let me know in the comments below. Tertiary structure. After seeing the formation of super-secondary structures, it can be difficult to draw a clear separation between secondary and tertiary. Nevertheless, at this stage, we can start thinking of proteins as fibrous or fibrous, like we see with collagen, as well as globular, which typical example is myoglobin. Additionally, this is where the R groups or side chains come into play the most. I'm going to call them fibrous. Now, fibrous proteins have an overall shape like a long rod, and the secondary structure also provides most of the information about the tertiary structure, since the helical backbone doesn't fold back on itself. And the only important aspect of the tertiary structure that is not specified by the secondary structure is really the arrangement of the atoms in the side chains. For a globular protein, it is necessary to determine in which ways the helical and pleated sheets fold back on each other, as well as the position of the atoms in the side chains. This brings us to an important question we need to ask, and that is, what forces and interactions are driving this higher level structure in proteins? So we've seen covalent bonding at the primary structural level, hydrogen bonding at the secondary structural level, and at the tertiary and quaternary structural levels, we're going to see a mix of forces and interactions that provide the overall conformational shape of our three-dimensional protein. We'll see hydrogen bonding between R groups of amino acids, hydrophobic interactions that cluster nonpolar regions into the interior of the protein, electrostatic attractions or interactions between oppositely charged groups, even side groups complexed or bound to a metal ion, as well as disulfide bonds which covalently link side chains of cysteines together. All these forces and interactions orient and restrict the possible folding patterns available to a polypeptide chain. Of course, not every protein is going to exhibit all these structural interactions, but they are features that influence their native state. Building from that, we can identify quaternary structure in many of our proteins, and this level refers to proteins that consist of more than one polypeptide chain, with each chain being called a subunit. The number of chains or subunits can range from 2 to more than a dozen, and they can be the same or different. You often hear these higher structural level proteins being referred to as dimers, trimers, or tetramers, and these just refer to the number of subunits they have, or simply they'll just be called oligomers. Now, these subunits will interact with each other non-covalently through electrostatic attractions, hydrogen bonds, and hydrophobic interactions. A couple of examples of proteins with quaternary structure are notably hemoglobin and, of course, ribosomes. Now that you've learned the different structures of proteins and understand how amino acid sequences ultimately lead to the native protein, you should now be able to imagine how it's possible to predict protein structure based on their amino acid sequence. This is made especially possible through the help of computers, which ultimately opened up different fields such as bioinformatics and computational biology. By understanding the flow of information in cells, we can even predict protein structure based on nucleic acid sequence. But we'll discuss more of that in later videos. So that about wraps things up for this video. I'll be making more content going deeper into some of the topics we mentioned today, as well as exploring all the rest of biochemistry. Thanks for watching.